In this last video, we're going to talk a little bit about those antibodies. So on our B cells, so here's our cell, we have these little projections that are coming off of it. These are the antibodies. These are proteins. And it's at the end of these proteins, remember at the end of this little thing, at the end of this little thing, that's where that BCR is, that B cell receptor that can match up with our epitope of our antigen. Once these antibodies match up with an antigen, they detach from the B cell. And then they can also form other different structures. And actually some of them can come off even before they've actually found an antigen. A B cell is just going to keep creating lots and lots of antibodies, and then they're going to roam around the body and they're going to see if they can find the antigens. There are 5 different types. However, in table 12.4 you can see all 5 types. You are just responsible for knowing these 3. The IgG, the IgA, and the IgM. If you remember genetics when you talked about blood typing, that I, that's the same thing that stands for immunoglobulin. So immuno means immune system, globin means globular, like a protein. So of these 3 types, you need to know what are their different structures. So G's only come in what's called a monomer. That means a monomer is essentially a single Y structure. So each of these that I've drawn over here are monomers. IgAs can come in monomers, meaning that they have a single Y. They can roam around as single Ys, or they can double up as teams. And when you have a team of 2, you have what are known as dimers. So this is when you have 2 of those Ys together. The immunoglobulin M's, they also come in monomers, but they can group up in 5s, and that would be a pentameter. So if you have 5 of them together, you notice that they make sort of a pentagon-like shape. I'm not very good at drawing them. Almost looks a little bit like a snowflake. Notice that the BCR portion is always facing outward. If you put the BCR at this side, you would never interact. So remember, the BCR, the part that's going to be looking for the antigen, is this little portion that's at the very end of each of these. So when you go around as these big clusters, you can touch a little bit more things than if you're just roaming around as a single. The other thing that you need to know about each of these antibodies is what part of the elimination process are they good at. So in the last section, we talked about 3 different ways that B cells will eliminate things. One of those was neutralization. So the ones that are very good at neutralizing are the G and the A. M does a decent job, but it's not one of its main things it does to eliminate. Those that are going to start the complement activation, that will start the classical complement protein cascade. The ones that do that best are the G and the M. We also have opsonization. So we talked about that. The ones that are, the G is pretty much the only one that is strong in this. And then lastly, we have that agglutination or precipitation, that increased phagocytosis. The M antibody is the one that predominantly will instigate more phagocytosis. So you notice that the IgG, the immunoglobulin G antibodies, the reason that they're so good at so many things is because these are the most predominant ones that are in your system. You also have a good number of A's and M's. And then you notice the other 2 that we're not even talking about, the E and D, Those are just so rare and so little known that I'm not going to have you learn anything about them. So what type of antibody you are will determine typically how you will go about eliminating the antigen.